Dear friends, the paper is the tissue culture, biological technique and developmental botany. Today, they discuss the topic in cryopreservation. In this topic, mainly 5 subtopic is necessary. One is the introduction, second one is the difficulties of cryopreservation, third one is the method of cryopreservation, fourth one is a gene bank, pollen bank, germ blossom bank, fifth one is the achievement made through cryopreservation. First, the introduction of the cryopreservation. Literally, the cryopreservation term is from 3 words. One is a cryos, it means cold. Per, it means save. Third one is the saver, it means to keep. Hence, cold storage before use, or simply the material or the living specimen is stored in liquid nitrogen under minus 196 degree centigrade. Here the introduction of the cryopreservation due to the rare and endangered plants is a gradual disappearance. Hence the save or the conservation is necessary. You know, the rare plants, endangered means once it's a cosmopolitan industry, it gradually disappear and the number of the plant is decreasing. That's why these plants is called the endangered plants or the endemic condition. The plant endangered condition plant saved with the help of the cryopreservation. It is not possible in the normal conventional method or the normal conventional technique because it is not stored for very long period. Main reason, the seeds are short-lived time. For example, the citrus, oil palm and rubber like plant is a short-lived seeds. It's not stored for very long period. That's why the cryopreservation technique is used for a very long period storage, even 1,000 years. Hence, it is a branch of the biotechnology or the tissue culture. It is called a cryobiology. This technique is called a cryopreservation. Now, the second one is the difficulties of the cryopreservation. If you want a cryopreserve, it's stored for very long period. For example, it is stored in the freezer. The main reason, the cells are vacuolated nature, larger in size, inside the abundance of the water. Once it is stored for very long period, then initially the cell wall is disintegrated and disappear, and also it's gradually disappear in the cell wall nature. Then cytoplasmic substance is gradually movement outside the cell portion. Hence, it's a very difficult, it is rupture of the cell wall, then dehydration of the water, then finally it producing a plasmolyzed condition. This process is called a plasmolysis. You know, the plasmolysis is continuously formed, the cell is a death in nature. That's why it's a very difficult to the preservation. Anyway, once it is preserved, then it is used for very long period, even 1,000 years. The methods of the cryopreservation. The first one is the selection of the plant. Second one is the addition of the cryopreservant. Third one is the freezing the material. Fourth one is the maintenance of the liquid medium. Fifth one is the thawing. Then finally regeneration of the plantlets. The first one is the selection of the plant material. Here the vital role. The main reason, the selection is the very successful, very important one. Hence, the young plant is selected. The main reason, the young plant portions contain the meristematic regions. You know, the meristematic region is the active cell division, cell elongation portions. Hence, the meristematic region is available in young plant. Then, highly cytoplasmic in nature, cytoplasmic condition is very high. Then, the smaller in size of the cells, hence it is very essential use for the selection for the young plant. Next one is the addition of the cryopreservant. The cryopreservant is nothing, is simply some chemical substance is used for the purpose of the cryopreservation. Hence, this chemical substance is called a cryopreservant. Generally, sugar alcohol, glucose, alcohol substance, polyethylene glycol, polyethylene oxide, dextrans, amino acids, glycerin, and some sucrose is also used for the cryopreservation purpose. Hence, the substance is or the chemical is called a cryopreservant. You are artificially added for this cryopreservant chemical substance. Next one is the freezing the material. The freezing is nothing. The material is preserved, maintaining. It's generally the freezing the material is suddenly, and also it's initially it's maintaining at least minus 1 degree centigrade is necessary. Hence, the freeze the material is the start at least minus 1 degree centigrade level. Next one is the maintenance of the liquid medium. Here the cryopreserve the material, the maintenance is the very essential, because if you want any material is cryopreserved, at least minus 130 degree centigrade is necessary. But here it is possible the nitrogen is treated in liquid condition. It means the nitrogen is a chill nature. It is treated minus 196 degree centigrade. The nitrogen is converted to the liquid condition. Hence, it formed a liquid nitrogen. It is optimum and identical, and also the ideal for the purpose of cryopreservation. Next one is the thawing of the material. Thawing is nothing, the removing of the vials from a culture medium. It means you are maintaining a culture, the culture medium portions contain the vials. The vial is removed from the cryopreservation. Now the purpose of the regeneration of the plant material, this process is called a thawing. Thawing is nothing, it's removed from the vials. Now, washing and reculturing. Washing is the plant material or the specimen is washed properly. It is very essential one, because you know the material or the specimen is in addition of the cryopreservant. This cryopreservant material is sometimes it is a toxic effect. The toxic effect is producing a side effect. That's why the washing is very essential for the materials. The washing is gradually, sometimes in ordinary water, sometimes in distilled water. Then it is washed in some surface sterilization of the material also, because the surface sterilization with the help of the ethanol, calcium hypochlorite and sodium hypochlorite, the material, the surface is sterilized. Hence, the material is now ready to the inoculated. This condition is called a washing the process. After the washing, now it is reculture the plant material or recultured the cells. You know the reculture means already the culture is maintaining for the preservation purpose. Now the regeneration purpose, the plant is cultured or inoculated in the nutrient medium. The nutrient medium is generally contain macro and micronutrients, carbon source, vitamin source, growth regulators and organic supplements. First of all, the major elements, macronutrients, the elements which are required for a larger quantity for plant normal development and growth. These type of the elements is called a macro elements. For example, the carbon, hydrogen, oxygen, nitrogen, sulfur, phosphorus, calcium, potassium, magnesium, iron. It is a macronutrients, the elements which are required for a maximum quantity. Similarly, the micro elements is also necessary. The micro elements, the elements which are required for a minute quantity or the meager quantity for the plant normal development and growth. This type of the elements is called a micro elements or the micronutrients. For example, zinc, copper, molybdenum, chlorine, boron, like this is called a micro elements. The next one is the vitamin source. The vitamin B is the very important role, especially thiamine, nicotinic acid and riboflavin is the very important for the vitamins. Next one is the growth regulators. Various growth regulator is involved for the nutrient, especially the axin and cytokinin. The axin is used for divisions and growth. Similarly, the cytokinin is also used. One is used for the root initiation and another one is used for the shoot initiation and shoot formation. That's why this axin and cytokinin is very important growth regulator. It is very important things. Next one is the carbon source is the essential. The carbon source is generally a sucrose substance is glucose, fructose, maltose and like this it is used for the carbon source. Then it is also very important role for the organic supplements. The organic material is used for the supplement functions. It is, for example, the coconut milk, tomato juice and orange juice like the material is also used for a supplement. It is a additional benefit for the growth and nutrient medium. Now it is ready to produce a callus. The callus is generally mass of the cells or the group of the cells. It's generally the undifferentiated mass of the cells. Now the cell is divide and redivide, it producing undifferentiated mass of the cells. This mass of the cells is called a callus form. Now the callus is ready to produce. The next one step is the regeneration of the plantlets. Once the callus is produced, the callus is developed and formed a new initiation of the root and shoot formation with the help of the growth regulators. Now it formed into a plantlet. A plantlet is nothing, it's a seedling condition or small plant, it is called a plantlet. The plantlet is transferred to the normal environment or the surrounding environmental portion. This process is called a hardening process. Hardening is nothing, the plant is from the chill nature. The chill nature of the plant not directly transferred to the field portion. That's why gradually it is transferred one place to another place. It is called a hardening process because it's a AC lab, then it is transferred into shady places, then it is transferred into a greenhouse, then it is transferred into normal environment. This process is called a hardening process. Then the plant is servable to the normal environment or the surrounding environment. It is adjustable. This process is called a acclimatization. After the hardening, the plant is have the fitness for the living in surrounding environmental portion. This process is called the acclimatization. Next one is the gene bank, cell bank, pollen bank and germ blossom bank. Actually, the bank is the collection of the material. Here also, sometimes the cell or sometimes the pollen grains, sometimes in the germ blossom or even a gene is collected and also maintained with the help of the cryopreservation purpose. They are maintained, then it is also supplied for the various country, national level and international level. For example, IRRI is IRRI, International Rice Research Institute. They are maintaining for the various type of the cells, various type of the germ blossom, various type of the pollens, especially in rice level. They are distributed national and international level and also maintaining for the cells and pollen grains. Similarly, a pollen grains is also stored with the help of the cryopreservation. Even 1,000 years it is a store. This material is supplied for the national and international level. It is called a pollen bank. Hence the cell is preserved, the pollen is preserved, germ blossom is preserved, gene is preserved, then it is distributed to the others. Next one is what is the achievements made through the cryopreservation? Various achievements in the cryopreservation because it is a basic tool of the biotechnological aspects genetic engineering aspect or DNA technology and also modern tissue culture level. Because these cryopreserve the material is stored for the 1000 years. If you want any time it is distributed, but that it is also sometimes difficult, but anyway it is maintained for the very long period. For example, if you want, they are summarized some plants which portion is the cryopreservation is optimum, which portion is the ideal, which portion is the favorable for the cryopreservation purpose. For example, if you want a carrot plant cryopreserve, the root especially a cell is the cell line portion is used for the cryopreservation. Cell is used for cryopreservation in carrot plant. Similarly, if you want a mustard plant, the pollen grains is used for the cryopreservation. The pollen is maintained and also preserved, then it is distributed in mustard plant for the purpose of the cryopreservation. Suppose it may be a plant of sugar cane, the meristematic region is stored. Meristematic region is cryopreserved. This meristematic region is stored and distributed. Hence, the sugar cane plant meristematic region is used for the cryopreservation purpose. Suppose it may be vegetative portion is used for the cryopreservation. The potato plant, the vegetative body or the vegetative portion, for example, the eyepiece portions is used for the cryopreservation purpose. Hence, they are summarized which portion is used for the cryopreservation, which part is used, for example, for this part. So, what is the benefits of the cryopreservation? Sometimes short-lived seeds is preserved in conventional method. It's only for preserved for few years only or sometimes in few months only. But in this case of the cryopreservation is used for even 1000 years or 2000 years, it is used for the preserve the material. Hence, if you want the any portion, even suppose cell portion, suppose it may be a pollen portion or the vegetative portion may be stored. After a very long period, any portion is ready to produce a regeneration. It's a very important thing, very important aspects in biotechnological research. It is also very important for the somatic level, the somoclonal variation level also very essential role. The somoclonal variation is nothing. At the time of the cloning, it is a somatically or the vegetatively, the some variation is present in differ from the mother plants. The previously the scientist is ignored, it's not necessary. But after the very deep research, it is very essential for the somoclonal variation, because first initially one morphologically or phenotypically only some variation is present. After that, the scientist analyzing the variation not only in phenotypically, it is the variation is present in this case of a gene level, nucleus level and also a cell organelle level differences is analyzing. Sometimes this chromosomal segment also some changes is producing. For example, suppose some aberrations like it may be formed to the inversion, deficiency, deletion, translocation and substitution like this. Then result it formed to the aneuploidy and euploidy nature, 2n minus 1, 2n plus 1, 2n plus 2, 2n minus 2 like this it's also producing. Then the euploidy, aneuploidy is also formed, the chromosomal segment is changes, even the chromosome number is also changed in the somatic hybridization. At the same time, this cryopreservation technique is one of the recent advanced technology. It is used for conservation of the rare type of the plants. It is used for the endemic plant is the conservation and also maintenance and conservation level. It is the vital role in the cryopreservation purpose.